Today we're going to talk about DNA replication. So you'll need your notebook. This will be your title here. We're going to talk about how DNA replicates. You might have heard the word replication before. To replicate something means to copy it. So we're going to talk about DNA copying, why DNA needs to be copied, and how it gets copied. You'll see down below in this picture, right, we start with one strand. This is the whole process we're going to talk about. We end up with two strands. Okay, so this is DNA copying or replication. Sometimes you'll hear us talk about it as synthesis, DNA synthesis. Synthesis just means making, right? So making more DNA. So take a second and think about it. Why would we need to copy DNA? Go ahead and in that answer box, answer that question. Why would we need to copy it? It's okay if you're wrong. I just want you thinking about it. So here's why we need to copy DNA in order to make new cells, okay? So you can go ahead and write this down in your notebook. We need to copy DNA in order to make new cells. We've just got done talking about mitosis and meiosis, which is cell division, how we make new cells. We have to copy our DNA so that we have two copies of those chromosomes so that we can divide the cell and create new cells. You don't have to draw this picture. I just need you to write the words on the top. All right, next thing I need you to write down, DNA is copied during interphase. Remember when we talked about mitosis? PMAT are the stages. Interphase was this whole outside gray part, right? Oops, sorry about that. It takes, 80 to 90% of the cell cycle is interphase, okay? We talked about what the cell does during interphase, and you can see here, the cell grows, it makes DNA, that's DNA replication or DNA synthesis, that's what we're talking about now, and it grows again, okay? And this is all to prepare for mitosis, the rest of the stages, right? PMAT, prophase, metaphase, anaphase, telophase, where it's going to divide. All right, in order to copy DNA, we need the help of enzymes. Go ahead and write down enzyme, okay? What the heck's an enzyme? Maybe you've heard of an enzyme. An enzyme is a protein molecule. It can help build or tear apart molecules. So it's a helper. I like to think of it as a helper molecule. Enzymes always end in the three letters ASE, or oftentimes they do. We're going to learn about some enzymes: helicase, polymerase, ligase. All of those are enzymes, and notice they all end in ASE. On the right here, I have a picture of a little kid with a Lego tower or a Duplo tower, and he's got a bat in his hand. He's ready to destroy it, right? So enzymes can rip apart molecules, but they can also build them up. So an enzyme or a helper could also have built that big Lego tower. So how do we copy DNA? What are the steps? Okay, so write this down. So the first step, helicase. Look, it ends in ASE. It must be an enzyme, a helper molecule. Helicase separates the two strands of DNA. I like to think of it as a molecule that's kind of like a zipper. It unzips the DNA. So if you look at the picture over here, see how the two strands are separating? If this were a zipper, it would unzip those two strands of DNA. If you remember from our DNA unit, hydrogen bonds hold these bases together. So what it really does is it breaks the hydrogen bonds in between those bases. You'll notice it kind of looks like the word helix. That's what you can use to remember it, right? It undoes the helix, meaning it unzips the helix. After we have them unzipped, okay, you're just writing number 2 here because you already have number 1 written down. There's another enzyme, DNA polymerase. Hey, look, it ends in ASE. Adds new nucleotides to each strand of DNA. So it adds the new pieces to each strand. On the picture on the right here, okay, remember we have our strands unzipped. Each DNA molecule has to be made of 2 strands. So one is going to be this old strand from the old DNA molecule, and then we're going to build another one on top of it. Okay, so this will end up being one new strand of DNA, and this will be another one. So this big blue blob is the DNA polymerase. Remember we said nucleotides, they have a sugar, a phosphate, a base. They're the building blocks to build DNA, okay? So you can kind of think of it as like, let's say you were going to build a Lego house, okay? You have a Lego house, you split it in half, okay? And then you add new Legos to rebuild the other half of the house, okay? So here we're splitting the DNA molecule in half, and we're gonna add new pieces to make each half a whole again. All right, so number 3, if we look at this picture, right, it's unzipping, we're adding the new orange strand here, and eventually we're gonna add all the way up until we have 2 molecules, because this whole thing's gonna unzip, and we're gonna have added 2 new strands. So at the end, we have 2 identical copies of DNA, and I'll show you how this happens. But basically, we start with 1, and at the end, they're gonna be identical. You might be wondering, well, they're orange and blue at the end. Well, that's just to show you what's new and what's old on the DNA molecule. I'll show you why they're identical in a second, and it has to do with the letters, the bases, the A's, the T's, the C's, the G's. All right, so let's go through this process again. You don't have to write this down. If you would like to, you may. But remember we said the first step, DNA is unzipped by helicase, right? So I have my little scissors, my zipper. It's gonna cut these bonds, okay, in our DNA. So now we've got half a strand of DNA or 1 strand of DNA and another strand of DNA. Okay. So now we're gonna add new nucleotides, new letters, okay? So here come the letters, T, A, C. Notice they match up, okay? So the letters are gonna match up. So A is gonna go with T, T with A, G with C, A with T, C with G, and so on, okay? So they're just matching using the same rules that you already know. So let's take a look, okay? Here's our old strand before it split up, right? The red is the old part, the black is the new part. What do you notice about these 2 molecules? They're the same, right? This has a T and an A, this has an A and a T. This has a G and a C, this has a C and a G, right? They're reversed, but that doesn't matter. It doesn't matter if they're, it's G and C, and then on this side it's C and G, okay? They both have the same 2 strands. 1 strand of ATGAC, 1 strand of TACTG. So our original one and our new strands, right? So we make 2 identical DNA copies. Sometimes mistakes happen. So write this down. A mutation is a change in DNA. Okay, and when we're talking about a change in DNA, usually we're talking about a change in the base, so a change in the letter, okay? Kind of like if you're typing out a word and you misspell it, right? That would be kind of like a mutation. You use the wrong letter. DNA, that happens too. Sometimes it uses the wrong letter. So there's a bunch of different types. You don't need to write down the types. I just need you to know that it's a change. I'm going to tell you some different types of changes just so you know, but it won't be on the test, okay? So in this one, the C got changed to a T, okay? So there's one type of change, right? They used the wrong letter. Another one, they inserted letters. So see, there's a TAT. On accident, they inserted it in the middle there, and it's not supposed to be there. And then sometimes we get letters that are deleted on accident. Okay, think about typing a document. All of these things can happen, right? You can accidentally type an extra letter in a word. You can accidentally miss a letter in a word or delete a letter, or you could switch what letter you're supposed to be using. You do not need to write this down. Don't write this down. How often do these mutations happen? Typically, there's one mutation for every 1 million bases copied, okay? So it does a pretty good job. But remember, we have 6 billion bases, which means every time we copy DNA, that would mean that there are 6,000 mistakes happening, okay? So that's a significant amount. Guess what, okay? We have other enzymes that help proofread the DNA and fix any errors, okay? So your DNA has a built-in proofreading mechanism, okay? Like an autocorrect or a spell check, which helps cut down on those mistakes. Here's what I want you to write down. So what causes mutations? A couple different things. We can have mistakes from copying DNA. This is where a lot of them happen. We can also have mutations from radiation, right? When you go to the dentist and you get an x-ray, you have to put that vest over top of you to protect your organs from getting any mutations, right? Your DNA in any of those cells. Sunlight, okay? We know that sunlight can cause mutations, and mutations actually lead to cancer, okay? Sometimes, not all the time. Okay, cigarettes, right? We know cigarettes can cause mutations, and that can lead to cancer. Some cleaning products, some preservatives, some viruses, and some bacteria. So lots of different things in our environment can cause mistakes in DNA. Now, most of these mistakes can be fixed by our body. Sometimes they can't, but mutations are something that happens all the time. So that's it for this lesson, okay? I will see you guys on the next live session.